Thank you very much. I have to say it's a real honor to be talking with so many people who've contributed so much to structural biology, and I kind of hope to hold up the Rutgers structural biology moving forward in here to the future. It occurred to me as I was putting the slides together, I perhaps undersold solid state NMR a little. So 15 years, it looks like we've gone back actually more like 20. So on this first slide here, I have the first 10 years of solid state NMR protein structure determination. And I wanted to show this for a couple of reasons. The first is to sort of highlight the difference between structures like this that are more models based on a single restraint, basically. So these are aligned spectra that got a single angle that would tell you the orientation of a helix in a membrane versus what I would think of more as atomic resolution structures of proteins or fibrils, which is kind of what I think of when I think of protein structures. And also to highlight how quickly the field of solid state NMR moved away or through something like the structure of MLF, a tripeptide, which was a PNAS paper in 2002, all the way up to full protein domains, something like GB1 over the course of only 5 or 6 years. So how do you solve structures with solid state NMR? I know lots of people are familiar with the bigger techniques. So NMR has contributed something like 13,000 of the 183,000 structures. Stephen talked about at the beginning. Solid state NMR has only done 140. So I'm going to talk about half to 10 of them, I guess, today. Carefully curated to be the ones I helped with. But basically, the idea is the first thing you have to do is label your protein with isotopes that are NMR active. So in the case of proteins, that's usually N15 and 13C. We also do a little back labeling with deuterium to help with resolution. And then it's a 4-step process. You have to do the chemical shift assignments. As any student who has actually done this will tell you, this is often a multi-year process. And so one of the things we've worked hard on is speeding that up. They have a collection of restraints. And so I'll also, over the course of the structures I show today, kind of highlight the different ways we've done that and the benefits and advantages of those. And then you do a structure calculation. Everything I'm going to show today was done with restrained molecular dynamics and some refinement, maybe with additional restraints you add at the end. And the overall goal here is to go from primary sequence to tertiary structure. And so, and unlike AlphaFold, we actually collect some information in the middle. So hopefully can get a little more detail in places where homology modeling is not perfect. So what are the first set of structures? So here's kind of the first era of solid state NMR structures. And they're small protein domains. You've got ubiquitin, you've got GB1, which is a G protein binding, well, the G protein part, and SH3 domain from alpha spectrin. And so what I wanted to highlight here is that the way you go from this, which was the first structure of a protein ever solved by solid state NMR, and this is a bundle of 7, I think, of the 12 lowest energy structures, and you can see they just barely hold together. It's a very, very low resolution structure. By adding more restraints, more distances per atom, you can get something that looks like this ubiquitin structure. And then finally, when you got to the GB1 structures that Chad, my PhD advisor at Illinois, was calculating during my time there, you have a whole lot of restraints, and you actually can get a very clean backbone here. So the difference here is from a few restraints all the way up to a ridiculous number of restraints, 7,000 carbon-carbon distances. Now that was excessive. That was definitely more than was strictly necessary. But then you also had the angle restraints, which I'll talk about a little bit more. And in this case, there was 2 sets of angle restraints. There were ones that were empirically determined using the chemical shifts, and then ones that were experimentally determined using the vector angles, which resulted in this pretty structure. So my contribution to this field of solving the structure of GB1 over and over again came from nitrogen to carbon distances. And so I spent my PhD using the pulse sequence TDOR. It became a bit of a running joke. Anytime anybody wanted to say something, Andy would pipe up and talk about TDOR. The nice thing about TDOR is it provides the opportunity to measure precise nitrogen to carbon distances. And the fact that you're including nitrogen also offers the opportunity to look for intermolecular contacts. And I'll talk a little bit in a minute about why that wasn't so important to me. But what I wanted to highlight here is that with 700 distances instead of 7,000, we were able to get a much tighter backbone RMSD. And at the point where at 0.2 angstroms of backbone RMSD, there's not you can't really get a lot better using restrained molecular dynamics. There's always going to be a little bit of wiggle room. But the other thing that was really helpful, and one of the places I think solid-state NMR is well, one advantage of the old quote-unquote generation of carbon-detected solid-state NMR experiments is you get information about these side chains. So one of the things I was so very proud of about this structure was how nicely restrained the side chains were in addition. And so as you know from chemistry, that you get to do inside a protein structure, it's the side chains that are usually doing the chemistry. And so if you just kind of focus only on the backbone fold, but you don't know what the side chains are up to, you don't necessarily have a structure that's going to tell you all that you could learn. So by having these really tightly restrained restraints, we were able to get a really clean structure of GB1. And one of the reviewers of this paper says, I hope this is the last time I see GB1 from your group. And so a year later, Chad and I published another structure of GB1, this time the crystal packing, which was interesting. So these GB1 preparations were nanocrystalline. And one of the hardest parts in solving this structure was that the beta sheets on GB1 here, the four beta strands here, actually stacked in a long, super extended beta sheet inside the crystal structure. So there were just as many contacts between the beta 2 and the beta 4 here as there were for any of the normal quote unquote intra protein contacts. And so I took that annoying feature of my data analysis and turned it into another study where basically by mixing the labeling, having nitrogen on one molecule and carbon on the other, we were able to detect intermolecular distances, prove that the crystal packing in our sample was the same as what was determined by power diffraction, not what was seen in the crystal, a single X-ray crystal structure. And we're actually to get a crystal packing that was very similar using our NMR data to what was seen in the X-ray. All right, so that was all I would like a first kind of segment of solid-state NMR structures. But the title talked about proton detected, so I want to spend a minute talking about the transition in solid-state NMR from carbon detection to proton detection. Proton has the highest gyromagnetic ratio, the highest sensitivity in NMR, and we utilize that using cross-polarization in solid-state NMR always. So when we're detecting carbons initially, we always start on the protons to get that extra sensitivity. But if you can actually detect the protons again, you get an extra enhancement, something like 3/2 times gyromagnetic ratio. So since the gyromagnetic ratio of proton is 4 times higher, this is a big gain in sensitivity. So solution NMR has been doing this forever. And so the goal when I kind of started in on my PhD was to be able to do this in solid state. The problem with solid state is that a static proton spectrum of a small molecule, NAV here, is, as we like to joke, full of information, but very hard to extract, which is to say there's a lot of couplings here, but you can see they're all on top of each other and they result in a blob. And so as we spin at the magic angle here, 54.7, we're actually able to average out some of the interactions we don't like, namely maybe the dipolar coupling here, to get nice isotropic chemical shifts. And so here's a spectrum of the same small molecule taken here at Rutgers at 100 kilohertz. And you can see we're able to actually resolve all the different sites in NAV now. And so we've lost that dipolar coupling information. We're gonna have to add that back in with the pulse sequence, but we've gained its resolution. And so that's why I kind of became enamored with proton detection. And we wanted to point out here is that the advantage of solid state NMR is that it is not restricted by the size of the molecule. It's only restricted by the overall sensitivity you can eventually get. So basically, the idea here is that no matter how big the protein is, you have these small domains with say 60 residues, you have the fibril with 140, and you have a big membrane protein, 300 residues. They all give the same line widths because the line width depends on the order of the sample and how fast you're spinning. So if you have a well-ordered sample, it doesn't matter how big the protein is, you can get nice 1/10 of a part per million line widths, regardless of how big the protein is, which is a nice advantage moving forward as we move to higher field magnets and can actually get the sensitivity to study these bigger proteins. So here at Rutgers, downstairs, as Stephen pointed out, if we were all together in the conference room, we'd be sitting basically right on top of the 800 megahertz magnet here at Rutgers, where we have this probe that spins 0.7 millimeter rotors at 100 kilohertz, 100,000 times per second. Very, very, very fast. And as a result, we get to average out those proton couplings and get very sharp lines. And so you can see here our favorite protein GB1 again, the 40 kilohertz spectrum in red with the deuterated, and the 105 kilohertz spectrum in blue with the not deuterated, which is nice for the biologists. The people have to actually make the protein. You get very similar looking spectrum. And again, once you have nice looking spectra, you're able to do all the things I'm going to talk about for the proteins here. All right, so why do we want to do solid state NMR? Like I said, the 140 structures out of the 180,000 in the PDB. And I just showed you a bunch of the sort of first decade of solid state NMR, we were studying nanocrystals. But the goal has always been to study fibrils because they do not diffract well, and membrane proteins in lipid environments. And so my lab here at Rutgers is currently studying peripheral membrane proteins. So basically our goal is to look at the contacts between membranes, specifically their head groups, and proteins that bind them. And so I'll talk a little bit about that at the very end if I have time. But the first two sort of stories I wanted to go through are about a fibril and a membrane protein that we were able to solve the structures of using solid state NMR, and kind of have a little bit of thought about what we could think about as a result of those structures. So alpha-synuclein might not need a big introduction, but I'll have a slide here anyway. It's an intrinsically disordered protein found in neurons. Its membrane bound state is a helix. And it's important because it's the primary protein component of Lewy bodies, which is the pathological hallmark of Parkinson's disease. Mutations in that gene cause Parkinson's, but confusingly often can either increase or decrease the rate at which they form these fibrils, which resulted in the conversation leading to the fact that the oligomer state from this picture from Dobson is perhaps more toxic than the actual fibrils themselves. So why do you want to study the fibrils, Andy? Well, the fibrils template the monomeric synuclein, causing increased rates of fibrillization, also causing different isomorphs. And so Jean today is going to talk about her ongoing research into alpha-synuclein. And so what I am left to do is just introduce the structure we solved of those fibrils. I was sort of the middle of 3 generations of PhD students in Chad Reinster lab that solved that structure. So why did it take so long? Well, fibrils are extremely hard to study by NMR because they have repetitive primary and secondary structure. So the sequence goes over and over again, and the secondary structure goes over and over again. And so like I was talking about in the workflow, the first thing you have to do is assign it. And then the second thing you have to do is get restraints. And both of those things are hampered by this set of challenges. There's also varied dynamics in different regions. This proved very complicated in the synuclein structure. The strong intermolecular interface I was pointing out that I detected in GB1, the reason I was doing those experiments is because I wanted to do the same thing in synuclein. And then finally, Talos, the protein which we use to predict dihedral backbone angles based on chemical shifts, is an empirical database. So basically it takes assigned chemical shifts and assigned structures and tries to correlate them one-to-one so that you can determine angles based on chemical shifts. But the concern was that perhaps fibrils are such weird protein structures that maybe the empirical database would fall apart, wouldn't work well, and we would lose that tool. So which is why Chad and company were developing those experiments to measure the dihedral angles instead of just using chemical shifts. So the first thing here, something about the primary and secondary structure problems. So over here is one of the spectra from GB1 we used to collect those 7,000 distance restraints. So every single one of these dots is a correlation between 2 carbons in this 56 residue protein. And my first 0th year in graduate school, the first summer before I started, I spent the whole summer picking these. There were 10. I picked every single peak on every single one of those. By the end of the summer, I was convinced I did not want to be a scientist. I was like, why would anyone do this? Years later, I realized that they took the 0-year graduate student and gave him the most boring job anyone could possibly think of. But it was a wonderful experience to get to take those data and actually try to solve structures with them in the coming years. So I accept my ritual abuse of having to look at these spectra over and over again. But what I learned afterwards was that this is the prettiest carbon-carbon 2D that's ever existed in the world. And this is what the carbon-carbon 2D of a fibril looks like. And so you can see there's a lot of really sharp, pretty signals. Those are going to be strong restraints. And then there's a bunch of things that we affectionately refer to as perhaps schmutz. These are less easy to turn into restraints. And the problem with synuclein is that the core of the fibril, the NAC region, gives these really sharp signals, and the rest of the fibril gives these slightly broader ones. And in order to solve the full structure, you have to figure out what's going on with all of them. In addition, there are a million Ks, a million leucines in synuclein, and they're all right on top of each other. So that was a challenge. So what did the structure end up looking like? So I figured for a retrospective like this, I would go back, and I was reminded during a thesis defense a couple days ago, one of Gene Baum's students, of my own PhD thesis, which included, is the final chapter, 2 very not correct models of the fibrils of alpha-synuclein. And I knew they were wrong at the time. I put them in there because this is as far as I'd gotten. Like I said, I was the middle of 3 students. So there was a student, me, and then the student after me finished the structure. So we have the TALIS results here, which show, as expected, that alpha-synuclein is just kind of beta sheet all the way. But there's these annoying gaps where the structure doesn't want to be a beta sheet anymore. And so we were all thinking about a beta, which is this nice little hairpin. And we were all, basically the restraints we had was all beta sheet and about 4 nanometers across. And so I was working under attempting to figure out how to get alpha-synuclein to thread through a 4 nanometer wide EM image, while also following along with the restraints I saw. So basically there was some restraints I saw between 46 and 58, a very kind of clear salt bridge. And there were some restraints. I can't point at things because people are in the way. Anyway, I can't see my right side of my screen. There was also a really clear restraint between G93 and A69. And so I was trying to come up with models that fit this. G93 had to be next to A69, and 58 had to be next to 46. And I tried a bunch of things. I fell in love with this model. It's the right length. It has my 2 restraints. It is definitely not right. And it's definitely not right because it has sheets where there's supposed to be turns. And what happened eventually, it turned out, was that this K was not 58 at all. It was actually K80. And the other thing I will point out here, just for retrospectives, is that what I'm showing here are the mixed samples, which are proving that it's a parallel and register beta sheet. But there are 4 or 5 peaks here to alanine 78. And I never understood what was going on with these 2, could not figure it out. And so 5 years after I left, the research group finally published this structure, and it's a Greek key. And so the reason none of my nice beta serpentine structures were able to figure it out is because this ridiculous little turnabout thing was happening in the NAC region. And you can see if you go back to the TALOS, it was there all along. The TALOS was telling us something very weird was happening in this region of the protein. And sure enough, here it is. And that restraint that I never could figure out was alanine 78 to valine 48, which until the last year of my PhD, nobody thought was even part of the fibril core. It took Gemma Kamelis, a graduate student that was my contemporary, doing years of experiments to even figure out that this last strand was part of the structure region of the core of the fibril, partially because of this long unstructured loop that was in the way here beforehand. So we knew the N terminus was unstructured, we knew the C terminus was unstructured, but this unstructured loop in the middle was a surprise to a lot of people. And well, the advice here is that, well, science is hard and you have to trust your data, even when it's telling you something that has nothing to do with your preconceived notions. I guess the final thing to point out here is we've gotten to about 10 restraints per atom, which is sort of where you need to be to get good structures. The second thing I wanted to tell you about was this outer membrane protein G.
It's time to wrap up.
All right. So I will just point out that this was an interesting story where the crystal structure had crystal contacts, which was showing an extended beta barrel, whereas the solution NMR on my cells was showing not that. And what we were able to show with solid state NMR, again, a lot of carbon-carbon and a lot of proton-proton restraints, was that the crystal structure was in fact giving an artifact, and that the real membrane structure, which we could get with NMR, was able to show what the structure will look like in lipid bilayers as opposed to in a crystal. So the group now is studying these peripheral membrane proteins, and I want to show off this 100 kilohertz spectrum, which basically lets us assign in the solid state a protein we already assigned in solution NMR. So I really think that's the future of solid state NMR, taking solution NMR hand in hand with solid state NMR to be able to understand structures that are more complicated than what you could do with maybe solid state NMR on its own. So yeah, I like to acknowledge my group. I showed you a little bit of data from the FMP, and a majority of the data I showed here was taken while I was in Chad's group at the University of Illinois. And again, very happy to have been invited, and very fun to get to see the history of all the proteins here at Rutgers and in the world.